Disease activity in MS is very heterogeneous, and we still don't know what are the pathological bases of this phenomenon. Genetic data have provided important information regarding new biological pathways involved in the susceptibility to develop the disease. But to date, no Genome-Wide Association Study has been published investigating the role of the genetic variants in determining disease activity. So this is the aim of our study. We enrolled patients with relapsing remitting MS who were at the beginning of a first-line disease-modifying treatment and who had no previously treated with immunosuppressive or second-line drugs. We defined the NEDA status along a 2 years follow-up, and we remind that the NEDA status is defined as the absence of relapses, the absence of EDSS progression, and the absence of new or contrast-enhancing lesions at MRI examinations. You can see in the table the features of patients. We had 790 patients, and about the 80% was classified as having some signs of disease activity. Starting from our patients and these genetic variants, we performed quality controls both at a per marker and a per individual level, obtaining our final list of 778 patients and more than 600,000 of genetic variants, which underwent the GWAS. This is the Manhattan plot showing the results of the GWAS, and we can see here in chromosome 14 that we highlighted a region of this chromosome in which 2 SNPs passed the genome-wide significant association threshold, but also other SNPs in the same and other chromosomes passed the SOS threshold for a suggestive association. This is the list of our top 10 SNPs, with the 2 top SNPs showing a protective role towards the NEDA status. And looking at the genes involved in the first SNPs, we can see that maybe the CMA1 and the CTSG genes were possibly involved. So looking deeper into these genes, the first one, the CMA1, encodes a protein which is expressed in the mast cells and has a role in degradation of the matrix of mast cells and also in the secretion of vasoactive peptides. The second one, which is the CTSG, encodes for a protein which is found in the aderophyll granules of neutrophils and may be involved in the remodeling of the connective tissues at the sites of inflammation. So we remind that the role of mast cells has been explored in the past. Mast cells are a resident population in the brain, they reside in the meninges, and may have a role in regulating the blood-brain barrier. They have also been linked to MS as they were found in the demyelinating lesions of MS patients, but their role is still not clear. We know that may have a role in the dysregulation of the blood-brain barrier, linking to the passage of lymphocytes and other immune cells to the CNS, but they also may play a role at peripheral sites, triggering the activation of self-reactive T cells. So maybe they represent a new potential therapeutic target. We, to conclude, defined some genetic variants which were linked to the NEDA status at 2 years follow-up, and so which could have a protective role towards the disease activity. In particular, 2 variants which reached the genome-wide significance threshold, but also other SNPs which showed a suggestive association. Globally, our signal pointed out a possible function of mast cells and neutrophils in defining disease activity, and if confirmed in future studies, maybe this could be a new potential therapeutic target.